What is apoplast? Hello? What is
Apoplasm
apoplast?
through the cell, sir.
Not through the cell.
Cell
The
wall.
water, right. Water movement through the cell wall or intercellular space. So, after absorption by rootlets of the root, water move through the cell wall layer or intercellular space until Casparian stripe of endodermis. That is one pathway. Another pathway, the water movement through the simplest mechanism. In simplest movement through the, what is simplest?
Through the cytoplasm.
Through the cytoplasm stand, cytoplasmic stand or plasmodesmata. So anyway, finally it reached the xylem of the root, right? From the
Yes.
xylem of the root, it continuation toward the shoot xylem, leaf xylem. Okay, right, through apoplast movement, the water move in root system. Okay, no problem. Then how the root move upright direction? That means from the root to shoot. Where is shoot? Shoot aerial part above the root. So the water to reach shoot system and leaf any aerial part, it need to be move opposite to the gravitational force, right? Hello?
Yes, sir.
Right. To opposite to the gravitational force, there need some of the energy. So today topic is how the water move upright direction. So that process called as ascent sap. Ascent sap means the upward movement of water from the root to the top to the plant against the gravitational force. That is called ascent sap. So actually this ascent sap mainly depend on three aspects. One is strong cohesive force. Second one is adhesion force. Third one is transferation force. Let's discuss one by one. Just just wait. Change the. Let me share the PPT. Slide. PPT visible or not? Hello? SN
Visible,
tabs
sir.
up. Right. So SN tabs up. Right. So what is SN tabs up? Already told you SN tabs up.
Is a movement of water against gravitational force.
Right.
Upward movement of water.
SN tabs up. Simple upward movement of water from the root to the aerial part against gravitational force. So this process called as SN tabs up. Definition clear first? Hello?
Yes, sir.
Right. So this is first proposed by Dixon and Jolly scientist, and again this is put forward by Dixon in 1914 and 1924. And mainly this theory based on the following features, that is the strong cohesive force and adhesion force and transpiration force. These three. Actually, before that, so to prove the asymptotes of so many theories are put forwarded. They are root pressure theory and capillary theory, but they are not accepted. The most accepted theory, that is the Dixon-Joly theory, the Dixon-Joly cohesive adhesive force theory. So according to this theory, the mainly water movement with help of the cohesion force and with help of the adhesion force and transpiration force. First, let's discuss one by one. What is cohesion? What is cohesive force? The cohesive force, the force attraction called cohesion force, due to which they cannot easily separated from the each other. So here the force between water molecules, these water molecules to another water molecule, strong mutual force, that means strong attraction. That is called cohesive force. That means, for example, this is the, so tell me one thing, the where the water transported, the which part water conducted, hello,
To the xylem, sir.
right, xylem, especially in xylem vessels, especially xylem vessels. So for example, this is the xylem vessels. Are you follow marker? Hello?
Yes, sir.
So this is xylem vessels. In xylem vessels, let's change the color. So the blue color only water molecules, one water molecule to another water molecule. So actually water cannot separate it due to this force. Which force? Can you separate water? Hello?
No, sir.
No, because of the strong cohesion force. So these are the water molecules. These water molecules cannot separate it with help of the cohesion force. Right. The second force is adhesion force. Okay. So adhesion force, the attraction between the water molecules, okay, water molecules and wall of the xylem, wall of the xylem vessels, wall of the xylem vessels or xylem element. Xylem elements, nothing but actually xylem elements, tracheids, vessels, so they are all. But mostly the water conduction with help of the vessels. So I already told you this is the vessel wall, vessel wall, right? So between the water molecule, that is cohesion force, cannot separated water. Okay, water cannot separated to water molecule. But here, what is the end point? What is the end point here? Last water molecule attached to the where? Hello,
Xylem.
xylem vessels wall. Okay, just imagine if there is no force between the water molecule and vessel wall, xylem vessel wall, what happen? Is there any attachment with both?
No. No, no, sir.
No, that means easily separated from the xylem walls. But here do not separate it from the xylem vessel wall. Because here also between the xylem vessel wall and water molecules also, the required one, utilizing one force. That force is called, the force is called, which force?
S-
Hello?
Strong cohesive force.
That strong cohesive force only between water molecule to water molecule. Remember,
Addition, sir.
right. The water molecule, here water molecule, that is cohesion force. But the xylem wall. But here xylem wall, actually it is not a same molecule. Xylem wall, a different molecule, water different. But the force between different. So here the xylem wall, water molecule by adhesion force. Okay. For cohesion force, water cannot separate. By adhesive force, water separated from the wall. Then how it is move upright direction? Okay, one is clear. If water separated from each other cannot move first, cannot move any direction, not upright. If the water, if the water molecule, there is no attraction between the water molecules, the water cannot move in particular direction. And if there is no force between water molecule and wall, cannot move because there is no attraction. So anyway, do not separating the water molecule. Here the sum of the forces, two forces utilized. Then how it is move upright direction? Only move upright direction only. Already told you, this is the root. Already you know from the root upper plus simplus root to the xylem. From the xylem it toward the shoot xylem to the leaf xylem. How it is move upright direction? How it is move upright direction? The upright direction moves only one of the important force, that is transpiration pull force. Transpiration pull. Transpiration pull, the unbroken water column. The unbroken water column, how the formation of unbroken water column? By which force? Hello? Hello?
Transpiration could.
No, no. But first of all, unbroken water column. This unbroken, the here. This unbroken water column formed by which force? I am not asking about upright direction. Only the water column formation. Unbroken water column formation by the which forces? It is actually, this is the continuation rope appear as a continuation rope. If the rope pulled from the top, the entire rope will move upright direction. The first, before that, how the column formed? This column, hello, how this column formed, this water column? Unbroken water column. Water molecule cannot separated, water molecule wall layer cannot separated, and as a result formation of column. How it is happened, I am asking you. Hello?
Adhesion,
Right, adhesion force and
strong cohesive force.
strong cohesive force, both. The property of strong cohesive force and adhesive force, water column formed. These water column
Yes, sir.
pulled upright direction by transpiration pull. By transpiration pull, the third aspect. So the entire column will move the upright towards uprights. In plants, such pull generated by the process of transpiration. What is transpiration? What is transpiration? Hello? What is transpiration?
Removal of water
Right. Removal of water through the
through the stomach,
right
leaves.
through the stomata of leaf. That is called transpiration. For example, for example, just wait. Can you see this diagram?
Yes, sir.
Transpiration of water here. Okay. Is it clear first?
Yes, sir.
Right. For example, So this is the vessel. Right. For example, this is root, shoot, leaf. So the same random stand. And also stand. This is, for example, this here the stomata. For example, this is stomata. Change the color. So they are the water molecules. This water molecule column formed up to, we don't have enough time. That's why just understand, just draw only few water molecules. These water molecule, water molecule, water molecule from the root to shoot to leaf. So they are the, for example, from the leaf to root, for example, 1,000 water molecule there. This 1,000 water molecules column formed by adhesion force and cohesion force. But after completed, so this is place of only for the 100 molecule only. But in this, during transformation process from the 100, 10 molecules loss, 10 water molecule loss. Here the 10 water molecule, the gap formed. Actually, this is place enough for only 100 molecule, right? 100 water molecule. But during transformation process, 10 water molecule evaporated, right? Is there how here the tension created, the gap created for how many molecules? Hello? Out of 100, 10 loss. Then
Yes,
how
sir.
many? Then how many water molecule need to be observed?
90.
Actually, this is place for only 100. Remember clearly, 100. Out of 100, 10 molecule loss. 10 molecule. Then how many remain?
90, sir.
90 molecule remain. Then how many need to be added?
More 10.
Again 10. Then again 10. How they are observed?
By the transpiration pull, they are adding one by one, like 10, 10, 10.
Right. Actually, by the transformation pool, last 10 here, and here the gap formed. Just circle gap formed. So this gap fulfilled by the next water molecule. Next water molecule means below water molecule. So the below water molecules reach the gap, filled the gap. Then as the next water molecule fulfill the gap, the tension created, then automatically from the underground, 10 water molecules, 10 water molecules, absorption by root, absorption by root. And again, if the water molecules, for example, 5 water molecules moved, and again 5 water molecules, absorption by root. So this is called transpiration pool. Through transpiration pool, whatever may be, how many molecules are lost, again this molecule fulfilled by the root. Is it clear? Hello, any doubt in this? Hello? This is the column, root, root. For example, here, this is the first. They are the water molecules filled under up to the stomata, filled up to the stomata. Actually, just imagine You are completely full. Column. There is no gap, but we don't have enough time to fill up this. Just imagine like this. This is adhesion force between water molecule cohesion force, column form. Okay, column form. But later, for example, these three water molecules lost through the stomata, through the process of transpiration, right? The next water molecule three replaced here, and next three replaced here, next three replaced here, next three replaced here. These three replaced here, and again there is here the gap formed, and this gap fulfilled with the underground water. Is it clear?
Yes, sir.
Right. This is the continuation process. This is happened continuously. Okay.
Yes,
Is it
sir.
clear? Any doubt?
No, sir. No, sir.
Right. So we clearly about the SN types of the first SN types of upright direction. In upright direction, only the three force: strong cohesion between water molecule, strong adhesion between the water molecule column, sorry, between water molecule wall of the vessel, and transpiration pool. Transpiration pool, the loss of water through the stomata. That's it. So these
Yes.
three aspects very important to ascent of SAP, right? This ascent of SAP also important question. And transpiration. Just now we discussed transpiration. So transpiration means what is transpiration?
Removal of water from the
Right.
stomatas of leaf.
Loss of water from the stomata. This process called as transpiration. And the here given about the stomata structure. The stomata structure that is not important one, but anyway we will discuss in brief. After that, stomatal movement. After that, we will discuss stomatal movement also. The stomatal movement also very important, right? So first of all, if you take the stomata structure, let's shape the diagram. Is it visible to you?
Yes, sir.
Right. Actually, this is the stomata structure. Just anyway, just draw in draft. So in stomata, two guard cell like this. Here already given guard cells. Guard cells. Always remember the guard cell outside thin layer and inside the pore thick layer like this. Thick layer. And it is surrounded by subsidiary cells or auxiliary cells. These are the subsidiary cells or auxiliary cells. So this is about the stomata complex. This is not an important one, but anyway, remember it. Okay. So how the stomatal movement? Stomatal movement means how the stomata closed, how the stomata opened. That's it. How the stomata closed and how the stomata opened. Let's see the stomatal open and close. In most of the plants, stomata open during daytime, right? Hello?
Yes, sir.
Most of the stuff because the transpiration happened during daytime. That's why the stomata opened during daytime and transpiration not occurred in nighttime. So the stomata closed at nighttime, right? Let's see the mechanism of the stomatal movement, stomatal closing and open, right? First of all, this is the guard cells they're given. First, see the what happened during daytime. To preparing this, the K plus pumping theory proposed by Levitt. Levitt proposed K plus pumping pump theory to explain mechanism of opening and closing stomata. According to this theory, accumulation of K plus ion into the guard cell in guard cells, K plus ion, where the K plus actually not given here. So anyway, I will explain. K plus ions into God cell from the subsidiary cells occurs the presence of light. Occur the presence of light. For example, this is the God cell. These are the subsidiary cell surrounding. Already told you. The surrounding cells are subsidiary cells. What happened during daytime? K plus K plus during daytime K plus enter into the God cells. Presence of the light. This coupled with efflux of proton lead to increase of pH of the God cells. So yeah, the accumulation of K plus ion into the God cells is associated with passive influx of Cl minus. So thereby decreasing the water potential. So when the K plus influx into the God cells during daytime, daytime, during daytime, apart from the K plus, here chloride Cl minus also from equilibrium, Cl minus also influx into the guard cell. So thereby decreasing the water potential, right? The minus negative means, and the negative means decreasing water potential. Minus decreasing water potential. Already we discussed yesterday. Minus negative, decreasing water potential. Positive, increasing water potential. So water thereby the enter the guard cells, making them turgid. Just imagine here the only negative, right? In negative created in guard cell. What about subsidiary cells? Subsidiary cells no negative. Only water present. This is positive water. Positive value present. Now tell me water according to water potential, water movement from the high water potential to low water potential. Hello? In subsidiary place only plus positivity. Water potential value positive in subsidiary cell, but inside the guard cells negative. Now tell me water movement from subsidiary cell to the guard or guard cell to the subsidiary cell. Hello? Hello?
From plus to, no, from minus to plus, sir. From God cells to above plus.
Leave it first. We will discuss later. My question is, first, according to water potential, water movement, high water potential to low water potential, right? Is it clear first?
Yes, sir.
This statement clear?
Yes.
Right. Due to the composition of Cl minus, where the negativity present, inside the guard cell or inside the subsidiary cell?
Inside guard cells, sir.
Inside guard cell. Here the chloride Cl minus, a malate ion also minus, malate ion. Here the malate ion also minus. So here the malate ion minus, chloride minus. The both are minus negativity. But in subsidiary cell only place positive created, positive water potential. Now tell me water movement from subsidiary cell to the guard cell, because I already told you the water movement from high water potential to low water potential. Here the high water potential in subsidiary cell, low water potential guard cell, as a result, as a water movement through the guard cell, through the subsidiary cell to the guard cell. What happen if the water move into the guard cell? What happened? Just imagine this is guard cell closed. For example, this is the closed guard cells. if you add water to the guard cell, the water molecule increasing in guard cell. As a result, the pressure falls on the wall layers here. Pressure falls on wall layer and turgidity due to turgidity, automatically this closed stomata open. The force of the water because the water flow is increasing in guard cell. Automatically it open. Automatically open. Is it clear first? Hello?
Yes, sir.
Any doubt in this?
No, sir.
Right. Now closing mechanism opposite. Closing mechanism opposite. The closing mechanism means here at night what happened in the absence of the light. This is happened daytime presence of light due to the presence of light. But closer, this is closer by absence of light. The K plus and Cl minus both move out from the guard cells. The reverse order means here the K plus already deposited K plus and Cl minus both are goes outside, moves out. So due to which the water potential guard cells increase. That means negative cells, negative separated and molet also separated. Molet utilizing respiration. So molet also utilizing. So no negative. Cl minus move out. Molet utilizing in respiration process. There is no negative in the guard cell. And water start movement as a result from here. The actual in guard cells. Just wait. At night, the absence of light, K plus and Cl minus ions move out of the guard cell. Due to they are the moving out, the water potential of guard cells increase, and water start moving out them, leading to closure of stomata. So simple here. Just wait. So here the in subsidiary what happened? This is subsidiary cell malate out, Cl minus out. So in subsidiary cells, negative. But in guard cell, positive. Already positive cell H plus already present in this. So if you compare chloride, malate outside the guard cell, but in guard cell only pluses are there, positivity there. According to the water potential, water move high water potential to low water potential. Where is high water potential in guard cells and in subsidiary cell outside the guard cell? Hello? Just wait. The whiteboard clear? Hello?
Yes, sir.
This is opening of guard cell. subsidiary cells. Actually, what happened during daytime? K plus during daytime, chloride both influx into the guard cell. So here Cl minus K plus, the only always it is positive. Subsidiary cells always positive. And during light, what happens? Photosynthesis takes place, right? During photosynthesis, what is the end product of photosynthesis? This is day. This is day. Light, in light, presence of light. This is light, symbol light. So due to the light, what happened? Are you follow marker? Hello? Is it clear?
Yes, sir.
Right. So during daytime, photosynthesis takes place, right? What is the end product of photosynthesis? Carbohydrate starch form. Starch form. And it is hydrolyzed as a malate. Malate. Malate also anion. Cl minus also anion. Now gradually this Cl minus increasing and also malate formation also increasing. Minus negative and Cl minus negative. According to water potential, always water move from the high water potential, low water potential. Here the where is low water potential? Where is low water potential means where is the negative value in guard cell or and subsidiary cell
In God cells, sir.
in guard cells. That's why the water move from subsidiary to guard cell, right? Hello?
Yes, sir.
Right. While the water moving into the guard cells, automatically the the water the water pressure falls on this wall layer, like these wall layers, guard cell wall layer. As the pressure falls on guard cell wall layer, this guard cell stretches out. As the result, pore is open. This is daytime. What happened during nighttime? Automatically reverse. In nighttime, in nighttime, photosynthesis takes place in nighttime. Hello?
No, sir.
No photosynthesis takes place. Right. And also in reverse process, what happened? Closing. This is closing. Closing of stomata. Subsidiary cells. These are the subsidiary cells. Inside the guard cells, K plus Cl minus both moves out. So here Cl minus K plus. And another anion here, the malate. Malate also anion. This malate actually, this malate anion utilized. And if any remaining, this malate anion also move out. This malate also move out. So no negativity in guard cells. Only positivity created. As the negative moves out, only positive created in guard cell. So where the high water potential now? In guard cell or subsidiary cell? Where the high water potential? Hello? Hello?
In guard cell,
Right, in guard cell, high water potential. Where is low water potential?
subsidiary cell.
Yes.
Subsidiary cell. Now tell me the water movement.
From guard cell to subsidiary cell.
Right. What happened if the water move from guard cell to the outside? Subsidiary cell. Is there any turgor pressure? If the water move out,
No.
no turgor pressure. As a result, without turgor, no, there is no turgor pressure. Automatically, the guard cells wall gets closer. Gets closer. That's when the stomata closed, like this. So this is happened during night time. Is it clear? Is it okay?
Yes, sir. We can
Right.
start unit 2.
Right. So stomata closing and opening also completed. Now the another important topic is mineral, second unit mineral nutrition. So remember the mineral nutrition. In mineral nutrition, just wait. Right. So the mineral nutrition mainly uh, essential elements. These essential elements, macronutrients and micronutrients. Just remember this all micro and macronutrients. If the macronutrients are, if the macronutrients are, first of all, where are the minerals? Hello? Where the availability of minerals? These
In
minerals
soil, sir.
present in where?
Soil.
Right, in soil, dissolved in water, right? So in this different type of minerals, they are the divided by R into two categories: macro, which are useful in high amount, and if it is low amount, it's micro. First, let's discuss about macronutrients. In macronutrients, nitrogen, if we have book, open page number 16.
Yes, sir.
All, right, all of you open page number 16.
Nitrogen, phosphorus, potassium,
Right, nitrogen, phosphorus, potassium, and
sulfur,
remember it, NPK, and sulfur, magnesium,
calcium.
calcium. These are all
Macronutrients.
macro. How many macro? How many macro according to Arnold?
6 are there, sir.
6.
Yes.
And micronutrient? Micronutrients are iron,
Iron,
manganese,
magnesium.
molybdenum,
Yes. Zinc,
copper, right?
copper.
Zinc,
Copper,
copper, boron,
boron,
chlorine, nickel. Actually, there are 16 essential elements. In 16 essential element, 7 macro and 9 micro. So anyway, let's discuss what are given in our book. So first of all, most important and macronutrient nitrogen. First in this, just you focus on nitrogen availability. First point, how the nitrogen available to the plant, number one. Second one, how the nitrogen utilized into the plant body. And where it is constituent, where it is constituent part of the plant. These three important. First, how they available, how it is useful, the where it is in constituent. That means where it is present in the plant body. These three important. So let me explain briefly here. First of all, nitrogen. All we know, where is nitrogen? Hello? Where is nitrogen?
Air.
Yeah, actually, the nitrogen gas, almost 76 to 78% is present in atmosphere.
Yes, sir.
But actually, in the beginning, I told you the minerals are present in soil, but not air. But of course, the nitrogen presents in atmosphere air, but the plants does not utilize directly the atmospheric nitrogen. When it is converted into nitrate or ammonia or nitrite, that form only received by plants. The first of all, atmospheric nitrogen, this atmospheric nitrogen convert into nitrate or ammonia or nitrite. After conversion, then only the plants observe. So that is the availability. Okay. Actually, atmospheric nitrogen, when it is converted into ammonia or nitrate or nitrite, then the plants can observe. Is it okay?
Yes, sir.
So actually, this atmospheric nitrogen can convert into ammonia by, as you know, that is from the rhizobium bacterium. Rhizobium bacterium from leguminous plant. What
Yes.
happened? Rhizobium bacterium root nodules formed, atmospheric nitrogen developed into ammonium. That's it. Another one is by physical mechanism. In physical mechanism means by thundering and raining season, automatic atmospheric nitrogen convert into nitrate. So anyway, by physical method or biological method, after convert into ammonium or nitrate, now this form of the nitrate, of this form of nitrogen absorbed by the plants. That is the availability, that is one clear. Now, where it is constituent, that means where nitrogen, actually this nitrogen constituent like amino acid, proteins, coenzyme, nucleic acids. If you take amino acid, if you take protein, the protein, what is the building block of protein? Hello? Building block of protein. Building block of protein, amino acids.
Amino acids.
Amino acid means NH2. What is present in amino acid? Nitrogen. The coenzymes, enzymes also proteins. Nucleic acid. What are the nucleic acid? Hello?
Again, they are nitrogens.
No, I am asking nucleic acids. What are the nucleic acids? Nucleic acids. Atmospheric nitrogen converting by ammonia, nitrate, nitrite. So anyway, this form by utilizing the roots by plant. Actually, in plant body, this nitrogen N presence in protein. Protein that means amino acid NH2 amino acid present. And another one is uh, acids, nucleic acid. Nucleic acid means DNA or RNA. If you take DNA, what are present in DNA? Nitrogen bases first. Nitrogen bases are adenine or thymine, adenine, guanine, cytosine, like this. Cytosine, guanine. So these are the nitrogen bases. In nitrogen bases, nitrogen is the main part. So that is. And also it is present in enzymes. And what happened if the nitrogen loss? And also this nitrogen present in chlorophyll. Do you know chlorophyll structure? Magnesium center, ferrous rings. Ferrous and nitrogen also present in this. So, right. Get the chlorophyll unique to the plant kingdom as in its molecule four nitrogen atoms. Sorry, four nitrogen atoms. The chlorophyll four nitrogen atoms. Nitrogen plays an important role in metabolism of plants. So that is the another constitution part of the nitrogen. So if deficiency of nitrogen, what happens? The loss of nitrogen, first of all, chlorophyll malformation. As a result, chlorosis occur. Because there is no nitrogen, there is no formation of chlorophyll. So that's why the chlorosis occurs. That is number one. And also, the plant shows stunted growth. There is no growth because actually, if you if you take any growth, any organism need proteins. If there is no proteins, there is no amino acid, no protein. No protein, there is no growth of the plant. So that is the deficiency character. And another one is, just wait. Right. Almost the similar deficiency characters. Next one, what is given after nitrogen? Phosphorus. So the phosphorus, the directly from the soil, phosphorus can absorb the plant direct from the soil. But where it is constituted what? If you take the phosphorus, DNA, phosphorus present in DNA, that is nitrogen base attached to the phosphorus. And also it is present in NADP, phosphorus, ATP, phosphorus, like this, and also RNA. So this is the constituent, mainly constituents part of the plant. And deficiency of phosphorus results stunted growth, same. Here also same, like a nitrogen, the same result here, stunted growth form. And plants green in color, but contrast nitrogen deficiency. Of course, in the both are same. The both, if the nitrogen loss, phosphorus loss, stunted growth appear. But in nitrogen loss, the leaves appear in green color because of chlorosis. But here, even it is the stunted growth, the leaf appear in green in color. Right, this is the main deficiency factor of the phosphorus. And next one, potassium, K plus. Just now we discussed the K plus mainly involved in movement of water, as a result, opening of stomata. Closing opening of stomata by potassium. Right? So where the potassium, how the potassium availability water, the potassium ions present in the soil, from the soil, the this water from the soil, the roots absorb. Then where it is constituent part, actually potassium it is constituent part of the enzymes. Mainly it is constituent part of the enzymes. And what happens if the potassium loss, the mainly uh, the potassium plays a key role in the opening and closing of stomata, already told you. According to the modern concept, the transport of K plus into the guard cells is an active mechanism involving the ATP. This element is also redistributed from the nature part of the young parts. So the deficiency symptoms are first noticeable in the mature leaf. of the lower region of the plant. In some legume, this deficiency result in white patches on leaves, stem. Such plants are weak. The cells in pith region tend to be get broken. So these are all the deficiency symptoms of potassium. First of all, actually, I will send the simple notes about the deficiency symptoms later. I will give you tomorrow.
Okay,
That
sir.
will tomorrow practical class. Please collect the material of deficiency symptoms of macro and micro.
Okay,
I
sir.
will give you a simple chart. Just follow the simple chart. That is enough to you because they are given some of the complexity. It is in time. It is not enough to explain. That's why I will clearly give you short notes. I will give you tomorrow. Just collect it from the class, from practical class. Okay.
Okay, sir. Okay.